to develop sustainable and climate resilient alternatives to conventional plant-based oils. Through the optimization of microbial production systems, microorganisms can be engineered to efficiently produce lipids with potential applications in the food industry, contributing to a more sustainable and circular bioeconomy. It is now my great pleasure to introduce our distinguished speaker, Professor Dr. Thomas Brück from the Technical University of Munich, Germany. Professor Brück is a distinguished researcher with extensive expertise in biotechnology, metabolic engineering, and microbial production systems. His research focuses on developing sustainable and biotechnological processes, including the production of microbial lipids and other valuable bioproducts, with a strong emphasis on resource efficiency and the transition towards a circular bioeconomy. Good afternoon, Professor Brück. We are very delighted to have you here, and thank you so very much for joining us, and we look forward to your insightful lecture. And now, to guide us through today's session, including the lecture and discussion, I would like now to hand over the session to our moderator, Dr. Lucia Diantika, a lecturer from the Faculty of Agricultural Technology. Dr. Lucia Diantika completed her doctoral studies at the Technical University of Munich, Germany. Her expertise lies in food biotechnology and microbiology, with research interests including molecular techniques and enzyme applications. Without further ado, I would like to invite Dr. Lucia Diantika to take the microphone.
Thank you very much for most of the ceremony. Good afternoon, ladies and gentlemen. It is my pleasure to welcome you to our guest lecturer, Professor Thomas Brück. He is my mentor and
PPT-nya kamu share, lupa di share screen biar nanti ke record. Iya, tetap di share screen biar ke record aja sih. Terus habis itu tinggal di slide biasa. Zooming aja nanti aja kameranya pakai kita.
Yeah, all right. I would like to give CV for Professor Brück. Professor Dr. Thomas Brück completed his PhD in protein biochemistry in 2002 at Imperial College and Greenwich University, London. And since 2011, Professor Brück has been tenured professor at the University Munich, Technical University of Munich, Germany, where he holds prestigious Werner Simon Chair of Scientific Biotechnology and served as director of the Thun Algae Tech Center. And also he is Vice Dean for Research and Innovation at the Thun School of Natural Science, and also contributes to national bioeconomy strategy as a member of Germany National Bioeconomy Council. His research focuses on the development of sustainable biocatalytic process and microbial production system, enabling the conversion of greenhouse gases and biogenic residue stream to high-value product for the food, chemical, and pharmaceutical industry. Today, he will give us lecture here entitled "Toward a Circular Bioeconomy: Microbial Oil as a Sustainable Climate Resilient Alternative for Cocoa and Palm Oil in the Food Industry." Okay, maybe the lecture will be around 30 to 45 minutes afterward. Okay, longer. Okay. So, and afterward, we would like to have a discussion session, maybe 15 to 30 minutes. It depends on your question. So, please, student, prepare your question to him. Okay. Now, the time is yours, please.
Thank you very much for giving me the opportunity to interview today. I was actually thinking we had about an hour and a half. That's why we have quite a lot. Can I have a clicker? Pointer and clicker? Anything like this? Otherwise, you have to change it for the slides. So I'll talk to you about yeast oil. That's certainly something that was planned, but I also will expand to some other activities that we're doing in circular bioeconomy. Yeah, it's all about sustainability and how we build a system that is sustainable for climate operations. and biotechnology is at the heart of it. And if you go to the next slide, please. So let me give you a little overview of what TUM is. Yeah, we are in the heart of Munich, so Bavaria, Germany. We are placed on one of all European universities. Yeah, that's excluding those from the UK because they're not in the EU anymore. We have 19 Nobel Prizes, and there's a cumulative grant funding of about £500 million each year that me and my colleagues we in through projects. So it's quite significant. We also the entrepreneurial university, which means we foster startups. So we have about 80 startups a year that come out of our research, and the size in 2025 is about 60,000 students, 720 professors, 12,000 employees. So we are really an size company, if you get. And we have 8 companies in Germany and 7 actually abroad. So we are a university that's centered in Germany, but actually being active internationally. So this slide said that we are a German university with a global mindset because we do important research. We are a sort of partner for other universities worldwide. And if you go to the next slide, you see how that plays out. So we are part of the European 9 universities, that's the excellent universities on the technical scale in Europe, including some like DTU, for example, in Denmark, or ETH3, or the EFL. Imperial College, where I studied in the UK, is also part of this. But we are internationally acting. You can see we have offices in San Francisco and Sao Paulo, in Beijing and Mumbai. But for you, most we're very active in Singapore. That's for over 20 years. Yeah. So TUM Create is our research arm, where I'm also active for 6 weeks a year. And there's also TUM Asia, which is a teaching arm of TUM, where you can actually apply for a master's and get a German degree. Yeah, not going very far away from your home. Then there's flagship partners. One that I interact with very thoroughly is University of Brisbane in Queensland, and also Tsinghua University in China. Yeah, so there's interactions there that are important for us because they're also good research that we want to leverage out. Yeah, next slide, please. So give you a little bit overview where I'm placed. I'm part of the School of Natural Sciences, which is a conglomerate of chemistry, biochemistry, and physics. Yeah, we were funded just 3 years ago under a major restructuring of the university, and our motto is discoveries at all scales, and it really brings together what we do. So we chemists, we look at molecules. The physicists actually look at the universe, or small atomic particles like neutrinos. Yeah, so there's black hole research, there's aeronautic science, all of that brings it together to really different scales. Next slide, please. And our foundations reach back to the beginnings of TUM after the Second World War, so 1957, actually, where there was the first neutron reactor in Germany for science. Yeah? And then in the 70s, my department was still the chemistry department. Still looks like this, so I'm in a very good, but they use it much newer. And around it, we built incrementally 5 different research centers with state-of-the-art equipment and research. Some of them, the last one is the Center for Protein Assemblies, where a lot of medical biotechnology is conducted and so on. So you can see here the foundation of the school, the 11th of January 2023, and next to me is actually the president of TUM, Thomas Hohlmann. So it's my namesake. He's also a chemist. He works on sensory science in the future. Next slide, please. So this is our campus. You can see on the middle there's still the old neutrino that's still active, but there's a lot of buildings going around. There's a lot of new buildings that are coming. Next slide, please. So, 3 departments, about 4,000 students that we house. We have 5 bachelor programs and 12 master programs. So it's quite a vibrant department with changing infrastructure strategies. The night school entails 90 professors, about 1,100 doctoral students each year, and over 1,200 employees. Next slide. And our missions is really to explore, understand, and predict nature's phenomena, really explaining nature along the trajectory of discovery. Next slide. And the vision is to combine physical, chemical, and biological concepts, bridging the engineering approaches to create solutions for fundamental societal challenges and educate the next generation. And the second to last, the crucial societal challenges, that is looking at sustainable food web, for example. And that's also the topic that I'll touch on. That's the professors that I work with. Usually when I write a proposal, there's usually an expert that complements my activities right in the department. That's really the power of the university. Next slide. Next to my role of being the Werner Siemens Chair of Synthetic Biotechnology, I'm also the Vice Dean of Research and Innovation, which means I have to coordinate the research of the 90 professors of the school and also look at how we can foster spin-offs out of the research that we do. So the innovation part is important. Next slide. And there are 6 main research topics that we touch upon. So accelerated scientific discovery is actually to do with a lot of computational simulation work that we do. While I'm a biotechnologist, there's another large research track on battery science, inorganic chemistry. Biomolecular engineering and design, this is a lot of tissue engineering, so we have a focal area on organoid research, which is important for health. Clean technology solutions, that's me, really looking at sustainable bioeconomies. And then fundamental tools, cost of evolution, we have a very large research cluster on cost-saving research, cost-saving physics, and fundamental science of health, that's biophysics, really looking at health systems and then quantum science technology. We have a large cluster on quantum computing in Munich, and that's not only combining the activities of Tom and the other university, the LMU, but also a lot of industry partners. So, for example, we have the quantum computing center of Microsoft. So they're working with us to drive this forward. Next slide. So a little bit about my chair. So we were really funding this chair in 2018 with money from the Werner Siemens Foundation to define a new area of biotechnology. And in my research group, we combine activities of bioparticles engineering, which is close to my heart. I'm an entomologist by training, bioinformatics, biological chemistry, bioprocess engineering, systems biotechnology as an interactive route to define new waste-free bioprocesses. That's really at the heart of what we do. Next slide. And really, because I work with enzymes, something I like best. We use 2 approaches. So one that uses either natural or engineered cells, where you can define a certain product outcome from an input. The input for us means agricultural waste. It's never glucose, very few times. It's always coming from a mixed resource that's either agricultural waste or aquatic waste, workable without. And in times where your product cannot be produced by a cell, that means either the metabolic pathway doesn't exist in nature, or most probably that product is toxic. Then we resort to circuitry processes where we arrange enzymatic cascades to transform the feedstock into an end product. one of those examples are alcohols, and this is bioethanol, you know, from as biofuel. It's also produced in large scale in Indonesia. But there are others, longer chain alcohols such as isobutanol, yeah, very toxic to the cell, very low concentrations. So you cannot make this product effectively using cell systems. So what you can do is you actually design cellular escape that converts your sugar into the end product, and therefore circumvent the limits of a natural cell. And that's usually the workflow that we carry out. So we start out with CO2 and light, and then come up with biomass that could be seagrass, algae, agricultural waste, cellulose waste, microalgae. We then apply our toolkit of cellular cell-free systems to generate intermediates, and that could be oils and fats, polymeric entities such as polymeric sugars or secondary metabolites, and things that are bioactive and have a very high value. And at the end, we refine those with our methodologies to produce food products, biofuels, bio-based materials, and bioactives. Next slide. Really, in my chair, there are 5 years of research that we follow up, and I'm trying to touch on some of them here. So one is microalgae, then oil yeast, filamentous fungi, actually to produce a lot of new enzymes that we're interested in. Designer cells, this is also something I'm talking about today, and cellular systems. So, next slide. So, first of all, before we touch upon the solutions, I want to focus you on some of the problems here. Okay, so global challenges. Next slide. Next. So, you have a little bit of German, something it's actually a cartoon from the newspaper. And that newspaper article was there 10 years ago. And it says on the left-hand side, climate change. And it says hi to that little man. And the little man says, oh, I'm sorry, I thought you were coming in a decade, not today. Yeah? And that's really the mindset of both people, population in general, and our policymakers, so government. They are not aware that climate change is now. You have these effects here. We have a very prominent now in Germany, which is a heat wave with over 40 degrees and drought. And in agricultural systems, that meant 7% of wheat harvest lost, 20% of rapeseed harvest lost because of drought. Something that nobody imagined will happen this fast. So we really need to keep this in mind. And as researchers, we are here to provide solutions and educate both the government and the people of our country. Next slide. So all of this taken together has certain effects. So we still have a rising population. There's a slide on the top left with 50% increase in population. You have this here very prominently in Indonesia, but this picture is actually from India, from the volcano. Yeah? These people want to eat. That means you need to provide more food, 70% more food by 2050. It's a huge challenge. Means you have to provide fertilizer, you have to provide land. It's an issue. Fertilizer particularly means a lot of CO2 emissions. Yeah, power wash process that you remind from basic science. They also, when they go from a trajectory of a basic society to a more developed society, as it happens in China, you need more energy, and that is 100% more energy that is produced mostly through fossil fuels, also fueling CO2 emissions. And all of that together will bring about climatic catastrophic changes. And I myself studied in Florida. I experienced 4 large hurricanes. Yeah. There were times where I didn't have water or electricity for 3 months, and that really formed my research. And that's actually a picture of Erica She visited me in 2004 in Brück. Oh, nice. Excellent. So I wanted to give you an overview on the lipids. Lipids are important for two things: food and also commodity chemicals. Firstly, it's food. We fry food with it, we put it in salad oil and so forth. So really inform advice it was 100%. At the moment, this trajectory is changing about 30 to 70 towards commodity goods, biofuels, polymer materials, oil chemicals. And here in Indonesia, your government made this decision to actually increase our production. That is really not a good decision in the long term. I'll show you why. Yeah, next. Of course, our production means you have to forest. You also have to change biodiversity. So the world could be just one iconic species of the touchstones. And that's for particular guidance in Indonesia, but it's very prominent, particularly if you go to foreign areas, which I experienced myself. So what we're trying to do is find alternative routes to apply these oils without impacting land use change. So no agricultural area use, no water resources use, no impact on biodiversity. And microbial oils can be a solution to this. But it's not easy. If you look at the slide, the yield of 1 hectare is about 4 tons of oil, and on the top of the productivity is your palm oil. It's pretty good when you look at land efficiency. It just gets worse if you go to the European equivalent: rapeseed, 3 hectares, sunflower, 4 hectares, and soybean in the Brazilian rainforest, 7 hectares. It's really inefficient, not good. Yeah? So we need to find something that's better using biotechnology. Next slide. And on top of that, we have to keep in mind that we need to act sustainably. And the guidance for that is these sustainability development goals that were defined by the UN some time ago. And the most important are goal 13, 14, 15, 12, and 6. These are the ones, if you look at the food system, are crucial. So I want to touch about 2 aspects that we are following. One heterotrophic by fermentation, one phototropic. Next slide. So the first is the oil yeast as an alternative to vegetable oils. If you look at this, on the top, there's some resources making oil. There's soy, flax, ricinols, and really in the plant seed, it's about 55% of oil. If you look at the ricinols plant, and it's produced in 200 days. It does get better if you look at the whole plant. It actually reduces the oil yield to about 8% if you calculate in leaves, stems, roots. Yeah, so if it's an annual plant, it's not really effective. But the yeast can do this in a week with a much higher efficiency, up to 80% or more. Yeah, just fermenting sugars that are left. Excellent. And our yeast that we're using, that we isolated 12 years ago, is Sotamotricious oleanosus. It's a non-conventional yeast. It's something that belongs to the whitewood farm. Yeah, I'm usually very hard to genetically manipulate, but it makes a lot of oil in a very short time, about 5 days normally. And it has a high tolerance against toxic products, which are important if you want to do fermentation on waste material, because usually the same those who have to process. And it's highly substrate resistant. You can use almost any sugar that you get, yeah, and other things. And you can use glycerol also. Next slide. And about 2019, I had a PhD student from Syria, my home country, He was almost finished, had 8 papers, already 3 patents working on this process, and then he came to me and said, sugars alone are not good. If I use acid, sugar, also known to produce oil, it's not good. Let's try those. And I said, well, you've already finished, try it. And he came up with this whole new process where you basically do a co-fermentation of sugar and organic acids. And then he devised a process to induce enzymes that can hydrolyze the cell of the yeast cell at the end. And then you just have to centrifuge to get rid of the water and separate it from the oil. And he didn't stop there. He also changed the fermentation conditions to produce different kinds of oils, and the recipe that he generated, he used to put back in the fermentation state absolutely. And with that, we actually had a very high publication and also an output. And now he's heading my startup. So, looking at this, on the very left, you have the cells with oil. So the oil droplet in the cell is stained red with the dye called red. Actually enters the cell, adheres to the outside of the oil droplet. And on the right-hand side is the literature that was out there published. Yeah, 0.66 grams per liter an hour. With the co-fermentation that Mahmoud developed, he ended up with a much higher productivity, 2.3 grams per liter. Significant. High biomass yields, 240 grams. This is really dense field fermentation technology. And 87% of oil, which was the world record. And if you take the culture out of the fermenter, it's actually so rich in oil it floats on the top. This is what happens there. And if you change fermentation temperature and oxygen content, you can produce different fats. You can produce either solid fat or liquid fat, something that is cocoa butter or palm oil, and something in between that is rapeseed. So you can actually define your fatty acids depending on the fermentation conditions without doing genetic manipulation, which is very important. Yeah, the food industry doesn't accept right now any genetically modified organisms. We don't do this very small anymore. In my setup, we actually have the Center for Industrial Biotechnology where we can do this up to 1,000 liters, and we generate master batches. So it's this fermentation system. We then centrifuge and we produce about 60 kilos of oil on the throw. Yeah, and that is then used to get industrial parts. Here you can have a view inside the slime reactor as this cultivation takes place. It is then pumped over, slide, and then processed via centrifuge. Next slide, and you can see this is actually a 3-way fermentation, classical centrifugation step that you use in the milk processing industry to separate the milk fat from the slime. All the equipment is standard. This is not something that is unusual. Yeah, so that makes industrial implementation easy. And you can see there's here oil coming out. Okay, so we could measure then also how we recover the oil using a fluorescent activated cells. So you can see when at the beginning you have these cells that are full of fat, they're all red, that's same. When you then apply hydrolysis, the cell wall is breaking up, the fat is congregated together, separating from the cell wall, and that's the sort of oil droplets that you get in the centrifuge. They all come together, and you can easily separate. Excellent. And you can follow this process of hydrolysis actually by capsuling, because the cells get smaller. Yeah, before they're very fat, and they break up, they're smaller, and they're scattered. It's actually measured. So about 24 hours, you're done with the process. You just leave it at room temperature, and it's done. Excellent. I'll just give you an overview of how we do this at a technical scale. So we produce the enzymes, we do the fermentation, and we do the downstream processes. And in the lab, we use the small centrifuge and the larger one. And the process goes a little bit like this. We do the fermentation, and then we actually incubate the filamentous fungi that produces the enzyme on the unresolved yeast. And then the filamentous fungus adapts to the sugars on the outside and releases just the hydrolase enzymes active on the cell. And this is how we produce the initial batch. And that we can then again measure using fluorometry. You have standard production systems. So we could have easy producer. After 48 hours, you can see that there's something to grow, and after about 200 hours, 7 days, the cells are completely digested. On the top right, that is the classical cultivation of Trichoderma residuals in bowls, and what we harvest is not the fungus, we harvest the medium that contains the enzymes. And if we deploy these enzymes on our yeast, they dissolve the yeast cells within 72 hours next. And once that has happened, we then apply centrifugation. You get 3 different phases. You get a water phase. The water phase itself, you can directly recycle in the next fermentation because it contains monomeric sugars that can be taken up by the yeast cells again as nutrients. The second phase, the yeast biomass residue, you use to make enzymes in the next round. And then you have the oil. So really, no waste water, which means it's also cheaper because you don't have to treat it. There are also some good properties of this oil. If you compare these two, so crude palm oil that is red, contains carotenoids, contains phospholipids, and these you usually have to remove by very harsh processes. You have to add benzoids, so activated clay, to remove both phospholipids and carotenoids, and then you have to do distillation to purify the triglyceride. We don't need to do this. It's almost 98% triglyceride that comes out of the centrifuge. That's why it's clear. It's also low in hydroperoxides, very low phospholipids for free fatty acids, high oxidative stability because they are less polyunsaturated fatty acids that can oxidize over a shorter time. And we can adjust fatty acid profile to mimic either palm or butter. The taste is neutral, there's no smell, and it has a longer shelf life. So for industry, there's also an advantage to use product. So when we go to our industry partners, we tell them, look, shorter fermentation cycle, we actually optimize to 3 days. High oil productivity, low energy demand, solvent-free. When you extract rapeseed oil, for example, you do hexane extraction. We don't need to do this, and it's waste energy. And we have 80% reduction in costs because of this. So we also did technical economic analysis for 10,000 ton facility and found that the return of investment is actually quite good with about 7 years that you have to invest. Yeah? Next slide. But we didn't stop there, so we wanted to diversify the product profile and add value to the classical triglyceride products. The first was to look at the distillation section. So sometimes we just do the steam distillation, and in the steam distillation, you found 2 products. Swelling. Swelling is very important for the prosthetic pharma industry. For example, it is the basis for packaging of mRNA vaccine, very highly fruitful. in the cosmetic industry, it's important for skin creams and very expensive. And here in Asia, most of the squalene still comes from deep sea shark. This is something that is forbidden in the EU. Our solution comes from waste olive oil, but that is very expensive. And on top of this, we have bigostrols, these are steroids, and what we can do with them, I can show you next slide. So, first off, we wanted to increase the swapping compound, because it's highly valuable and has really good economic features to the product. So we added an inhibitor of the biosynthetic pathway for soybean production. Teledafine. It's actually a chemical compound. You can buy it. And we did a clinical for this and then showed that it actually increases soybean production by inhibiting soybean oxidase. And with this, we looked at productivity. So you can still see there's only really 2 commercial products out there that are competitive. So charcoal oil obviously is a lot better, but it's zero. It's really damaging the environment. Amaranth oil from amaranth and grain is also very high in squalene, but we don't produce enough to meet the market demand. And then it's olive oil, you can see it's very small. So this is the only option we have in Europe. And our yeast oil without the inhibitor was almost on par with it. But when you add the inhibitor, it was better. And that really helped us to increase the value of our process. So this is what you can do with squalene. You can also produce squalane. Squalane is what is actually the compound used in cosmetics. And with the bigostrols, the steroids that are also contained in this fraction, you can just add light and you produce vitamin E2, also very highly valuable. So it's a way to actually produce high-value compounds from residues of your oils. And this is just an overview of how this is happening. Excellent. And the next was to upgrade the yeast oil fatty acid. And together with Dr. Loll, we actually found a new enzyme family called oleate hydroxylase. And that enzyme adds a hydroxyl group at a double bond in monounsaturated acids. And our yeast oil is very rich in oleic acid, delta-9, C18. So it actually opens the double bond and hydroxylates it, and that without a cofactor. So it's quite efficient. And you can see here there's almost a complete conversion. And the reductant product is 10-hydroxystearic acid. And that's both the viscosity modulated skin creams in the cosmetic industry as well as bionutrients. Yeah, highly important. The market value is about €200. So quite very. But we didn't stop there. We also thought, okay, how can we make this even better? So the oleate hydroxylase only reacts free fatty acids, but we haven't even tried this one. So the first step that we did is find a lipase enzyme that can keep off the fatty acids from the glycerol backbone. And normally that is an equilibrium reaction that's more towards the triglyceride than the free fatty acid. And we selected at the end lipase would do the reverse, actually produce free fatty acids. And with that, we then applied the oleate hydroxylase, and from there also went a step further with a secondary alcohol dehydrogenase that we isolated to produce 10-ketostearic acid. And that's a clinically important compound for the diagnostics. It's very, very valuable. Yeah, it's in the thousands. And we found that this works very well if you do it cell-free. So this is an example of enzyme cascades. You don't need a cell. you just add 3 different enzymes and you get a product in high yields. Yeah? You just need to have the right conditions. Now, a circular example for food industry and how we scale. So, where we wanted to go is produce oil butter with our yeast oil. We produce already chocolate and as cookies. So, in terms of application, we showed it worked. And most importantly, for you as food-related chemists, you can see this magic curve. Your monthly in chocolate is induced by a specific melting of cocoa butter, which happens at about 28 degrees. Yeah, if you don't have that very strong cut, no chocolate field, no fun. We can mimic this exactly without this. So it's quite good. Yes. So in Germany and in Europe, you know, we develop practice a little bit different from Indonesia. So what we did is we teamed up with a baker that produced a lot of waste. So usually the baker bakes in the morning, people come in, buy the bread, but in Europe, customers want to have all bread from the morning to the shop closes at 8, produces a lot of waste. So over a ton of waste was small baker in a week. So we took this bakery waste, which is still good to eat, it's just not as functional. We hydrolyzed and liquefied this bread using commercial baking systems that are already on the market, yeah, to free the glucose, so basically degrading the starch of the bread and also the amino acid, the proteins in there to nitrogen cells. We then did a fermentation. We produce yeast oil, applied our process, and then did another round of baking with the products. So it's circular use of waste stream in the urban environment. Works also good with yeast, with rice or any other cereal. This is just an overview of an automated bakery. You can see it's mostly completely automated. Auto production is calculated in the cost model to satisfy the customer. Excellent. And this is the waste that we produce. It's not so good to eat, but nobody would buy it. Yeah? And the bakers don't have a route to dispose. They actually have to pay people to take it away. And normally it is just used for making biogas. Biogas is really bad in terms of efficiency. Only 5% of the carbon you put in is converted into methane. The rest is sticky. Exciting. So what we did with ours is actually produce high-value confectionery products. So on the left is something what we call donuts. Then we have a tartlet, so they have the others fresh. And the gray stuff you see there is sort of break over to the next one. It's microalgae that we use for food. And these microalgae are very rich in proteins and vitamin B9, especially. So we could sell this under certain specific conditions, like we can't roll it out big into the market because yeast oil, even though it is exactly the same chemical as the palm oil or cocoa butter, it's a different process and it underlies regulatory conformity. So we have to apply as a company to new regulations in Europe. This is something you don't have in Indonesia. Yeah. And it slows down market constraints, the actual valorization of the sustainable product. This is something we need to change. Next slide. Now, a little bit on land use efficiency. I told you we need to be better than palm. And we did this approach. So, when you look at using 1 hectare of palm oil production, you replace crop monoculture. You have to imagine anything you do in agriculture is 2-dimensional. There's crop, there's field, and you only have certain mass efficiency for that crop. We are much better in that respect, and we can go vertical. So an industrial fermentation system is 300,000 liters, really big, something you see on the left-hand side. So with that, we can actually produce on 1 hectare as much oil as you would produce for 80,000 hectares palm oil monoculture. So we're better. And we're even more better if we look at our European solution. Rapeseed, as for example, you need to replace 240,000 hectares CO2 land. And you can use that land to replant forests, and you save a lot of CO2. Slide. And the CO2 we also quantified, so just for the German version, 1.7 million tons of waste spread a year in Germany. That can be converted to 800,000 tons of yeast oil. Yeah, the yeast breathers and heterotrophic organisms, there's CO2 coming out, but if you look into the savings of land, the forest, it's about 200,000 hectares. You come up with 16.8 million tons of CO2 that can be saved using this technology. So at that point, you're waste free and you save CO2. And that's really important if we think about giving the next generation a chance in sustainability. So we built a company, and that company is now viable for 5 years, and we're currently in industrial development of this process. So, as I said, now the alternative. Yeah, so yeast uses biomass and produces CO2. What about if we can just take CO2 out? And algae give us the opportunity to do that. Phototrophic organisms, yeah, photosynthesis inside. So what you need when you cultivate algae is sunlight, you need CO2, and you need water. Excellent. And to produce oils, yeah, and some remaining products. Excellent. So we calculated that. It's about 150,000 microalgae that still are open for research. And every time I go diving in Bali, I probably find a new. Yeah. So in industry, there are 10 to 15 microalgae that are in use. So there's far more that are out there that are important. And we work on some of them, yeah, that are faster in growth, so about 10 times faster than any industrial plant. And they produce 13 times more oil. So if you again look at rapeseed oil in scenario, you need 1.5 ton per year and 1 hectare. And with the same hectare of land, in the same year, it produced 19 tons of oil with algae. So far more efficient. Yeah? So if you look at barrels of oil, yeah, you need only 74 square meters. So that's quite efficient in terms of land use efficiency. Excellent. So algae, we know already there's certain products that are on the market, but they're still niche products. So it's omega-3 fatty acids, carotenoids, vitamins, polystyrol. It's mostly for the food industry, either as performance additives or as nutraceuticals, yeah, to improve your health. But it's nothing like really big product. Excellent. We wanted to change this and looked at certain algae that we could work with. Usually in my lab, we have a train of scaling. So we go to the environment, we then test it at high throughput manner in multi-type plates. We then have dedicated growth chambers, bioreactors, and then our algae tech center inside, where we can scale to industrial scale. And the algae tech center is actually also the Airbus campus in Ottobrunn, where we can simulate climate situation in other countries. Germany is all that good for algae. We have not enough sun, and the winters are really cold. So Indonesia is good, Malaysia is good, yeah, all the regions here and southern us up. And for us, we have the technology, we can actually scale our technologies in this architect center, and then without building a pilot plant, which is skip space, build a production plant in the dedicated region that we want to. Yeah. So we simulate light. So this is just an example. It's glass house, you have natural light coming in. The blue line is a varying day. And if you want to simulate California, for example, for population, you add LED light in real time. And how you do this is on the top of that building is a light system that actually reads out the current light every second. And then substitutes the artificial LED light to come up to the target region we want to scale. So it's quite unique. And also on the top, you have a lot of equipment to regulate humidity and temperature. And we have these flat panel cascading points up to 500 meters for scale. Next slide. So it's basically 3 different units with each about 500 square meters of research space. And you can see here there's various surface areas between 8 square meters and 50 square meters to control. Next slide. And 10 minutes left. Okay, and I'm quick. Next slide. We go through one more. So when you want to do food products, you only can use left-hand side, closed systems, to avoid contamination. Open points with the biofuels are not. Next slide. So we produce a lot of products in there, from supplements to omega-3s. Next slide. But what I want to show you is how you can apply algae in food systems. So there are 3 that we actually work on: Chlorella soloponia, freshwater algae, Dunyanella salina, which makes beta carotene, and Atrospina maxima, which is spirulina with purocyan. And we group them in our systems in the slide, and then generate food products that are rich in vitamin E and proteins. So again, tablet, again cake, some dumplings that have covering of beta carotene. So all of these, when you eat them, they improve your health. Yeah? And are good for the environment. And if you have something like on the right-hand side with phycocyanin, it's also fun to eat. It's a mousse with phycocyanin. If you illuminate it with white light, it's blue. If you illuminate it with black light, it's shiny red. So, quite fun. Okay, I will probably make 2 minutes on the terpenes. If you terpenes, you know, they're fragrance compounds, they're bioactives, and also carotene, of course, the terpene. But what we are doing is producing them recombinantly in cells, intermediate cells. And the reason is single components, constant quality, season independence, and scalability. And how we do this is we usually transfer genetic information from one organism to another. Usually that's colon. We then use waste biomass hydrolysis to feed these organisms and produce these very beautiful compounds that are bioactive. In this case, is either the train A, which is anti-inflammatory symptoms. Example. And to do that, you have to do up and downstream metabolic engineering in E. coli. This is something we do on a regular basis. Next slide. And there are 2 examples. I will only touch on one, on the nicotina tobacco cellulose. And next slide. So the reason why we produce those is we have an increase in pests with climate change. Yeah. So foreign pests actually eat up our plants, and then we have a harvest loss. In Europe, we also have another issue. The classical plant protection agents like neonicotinoids are phased out completely to protect the bee population. Yeah, so it's not about it. You have to find new routes. And we thought ourselves, okay, we want to produce plant protection, that's not killing the insect, because then you induce resistance over time. If you repel, so if you just drive the insect away, no resistance is induced this long protection period. Next slide. So there was an ecological background to do this, and the compound needed by the breakthrough. Next slide. So what we produce is beta-centrally low. This is the compound on the right-hand side from wheat bran, which is a million engineered. We engineered the biosynthetic pathway that came from the tobacco plant in Australia into E. coli to produce this compound. Next slide. So basically, this is what happened. You have a precursor of terpenoids acting in DMAP, generating the general precursor of D-terpenoid, general biphosphate, and then you have an enzyme that does the cyclization of that linear precursor to produce separately as two racemic compounds. Next. We then tested this, and the productivity was good in E. coli. Next slide. And we then devised a complete circular process where you take the residue from milling, so the wheat bran you hydrolyze, then add your E. coli to a fermenter, produce a compound, you test it on your target organism, in this case, leaf lice, and then you deploy it back in the field. So waste from the field is producing a compound going back to protecting the crop. And we did this in a 50-liter fermenter with an unusual application, because you can see on the left there's the pollen. This pollen contains amylase that selectively takes out your target compound centrally known and avoids toxicity effects over a long production time. And we put the cells back into the fermenter to produce our compound for a long time. We could then dilute it off the column and have a pure compound. We then tested the bioactivity and showed it really is not toxic either to insect cells nor human cells, but it had some antibiotic activity around negative bacteria. So it had also topical antibiotic activity, but on the middle you can see the leaf lice assay basically showing that the leaf lice did evolve once this compound is put on the plant. So it's a natural biodegradable insect for plants. Okay, we'll skip this and we go right because every day for 2 hours. All the way through the go to the summary. So okay, just a little bit. Okay. So just to give you an overview where we go with biotechnology. We did a lot of things by hand over the years. Now robotics enters our realm and things can go much faster. So in my lab, we invested a lot in robotic infrastructure. You can see here, the Bioeconomy-Microbial X7 can actually do all the assays that we usually do by hand by itself. And also parallel fermenters to increase our bioprocessing. Next slide. And what we do then is basically automate our research and combine it with AI. So we can actually run experiments faster and also get our results faster. I think this is the horizon. Next slide. And then we go to the summary. So we use also production systems to use waste streams and then toxic side streams. And the yeast is indirect CO2 conversion to value-added products such as yeast oil. Yeast lipids can replace plant oils, improved in chemical applications. You have a circular use of urban risk use, such as stale bread, reduced land use, improved biodiversity, CO2 savings, and the products have the same cost and physical performance as palm oil without the ecological implications. For the microalgae, there's a direct use of CO2 for production of value-added products and increased cost efficiency, and circular processes are a new road for equal and cost-efficient bioeconomy. Last slide. For the glycopenoids, sustainable scaling of high-value products such as plant protectants. You can have enzymes that access novel chemical product diversity, and then maybe the last like AI can accelerate process optimization and can solve a multidimensional research space, and robotics combined with AI is for biotechnology research. The future is really aimed at automated experimental design and conduction. So we as humans need to learn to work with computers, not to be afraid of them. We still need to supervise the research because only human intelligence can actually come up with ideas out of the box. ChatGPT is not this. And with that, I want to finish. Thank you.
Thank you very much.
Yeah,
Okay, please. Thank you very much, Professor Brück, for very inspiring presentation today. Now we have a very short time for a question and discussion session. Please, who has question, please raise your hand. Oh yes, please. I think you have also lecture, right? After this.
Okay, thank you, Professor Thomas, for the very interesting presentation. I'm Brück and I'm from Rio Province, the one of the largest province in produce palm oil. So palm oil is something I have experienced not as only as community, but also as part of the livelihood of many communities. And I have a personal interest in the future of palm oil and alternative oils, of course. And my question is, considering that substrate costs can be a major factor in microbial oil production, what do you think is the key to making microbial oils economically and environmentally competitive with sustainable produced palm oil at a large scale? And could agricultural residue or industrial waste play an important role in making this possible? And I love to understand the palm oil sectors, but I'm also genuinely curious about whether biotechnology can make our food system better. And the last, since you are coming from Germany, I'm also curious about the European perspective with the increasing emphasis in Europe on deforestation. Do you think the future is necessarily about replacing tropical crops such as palm oil with alternative technologies? Or could better traceability, certification, and improved production practices allow sustainability produced palm oil to remain part of the Europe food system? Thank you.
Multiple questions.
Yes.
Okay, so let's try to answer this. I'm aware that palm oil production is majorly carried out by small farmers in Indonesia and that they have been invested. We are not proposing to remove that. Yeah, I'm against expanding it. So you would have to be more efficient, and you also have to improve living conditions, obviously. Yeah. One solution that we talked about was actually using the residues of palm oil production, so the spent palm oil bunches as a sugar source. They're lignocellulose. You can actually degrade them with enzymes. Yeah. And you can use that then to do fermentation with yeast to produce more palm oil. Yeah. It's more efficient than just burning that residue on site, which is the common practice. Yeah. Certifications, I don't believe in, particularly not when there's financial interest. Yeah, my experience is even if you have NGO-driven certifications with the right kind of money, the company can buy anything. Okay, it's my personal experience. I really think that there needs to be an economic advantage for both the producer and the company that uses that oil to switch. Because imagine you have that waste that you accumulate with the corn palm oil production, and you convert it on site because you don't want to produce it somewhere else. Yeah, transport adds cost, and it's also emitting CO2. So you really should produce or install fermentation capacity on site next to a palm oil production, and train to operate. This is not rocket science. It's basically a process like growing beer, just a little bit different. And it's completely halal, by the way. So it's something you can implement here without a problem. But you need strong investment. Your farmer can't do this. It's a company that uses and finds the oil that needs to do this. Yeah. We've been talking to companies saying but it's very hard at the moment because the economic climate is hard. And in the EU, the restriction of palm oil is really evident. So we were approached by a large mineral oil company who produce biodiesel from palm oil because we have the admixture on it. So every diesel product in Europe needs to contain 7% of biodiesel, right? They don't get the resources. So palm oil was the cheapest resource and they're not having access. So they're interested in rolling this out. So my hope is that maybe from the end user that uses this, there is a push to the middleman, to the refiner, to implement this process. There's no benefit if we do this in Europe on a small scale when you can do it much bigger here. I hope that answers the question.
Thank you.
Okay, thank you very much. Regarding this palm oil, also have a question. Is it also any investigation of the AG composition, the acetyl composition of your oil? Then it can be claimed as similar to palm oil. Ah, okay. Okay. Okay, the question is. Okay.
Good afternoon, Mr. Thomas. I'm the alumni of from this department from the master degree, and now actually I'm working with the palm oil farmers to help them workshop about the cultivation and stuff. So I'm here to hear about your exciting topic, and I have some questions, but based on your website, that I already look over your website. That you have the company GS,
Yeah.
if I'm not mistaken, and I just curious about the future if you want to commercialize it, because I don't see that you already started the use, because I think about the regulations and stuff, and how to educate the potential customer about your products. And I saw that there's no drawbacks about your oil, and it can be a hard combination for us too as a palm oil producer. And yeah, I saw about your resume, it's interesting. And I just want to ask, like, how do you convince the potential customer? Thank you.
Yeah, so we already did customer trials. I mean, for the bakery goods in this stuff, also in the cosmetics and also in the industry. And there are bilateral collaborations between GST, my startup, and larger companies. So GST is not a dedicated producer of palm oil equivalents. It is a technology provider. So we provide the technology set, particularly to the infrastructure that's with the customer. For example, when you have a bread producer, most of the fermentation capacities to make the sourdough bread, for example. Yeah, you can retrofit these fermenters to make yeast oil using waste bread. We have several interactions with mineral oil companies already, and it is really the challenge to motivate them to test this process at industrial scale. You can imagine that one run of 100,000 liter fermentation is about 100,000 euros. And that is really not much money if you look at the context to test. But still, people like Dow, IBM, or Chevron are adamant that we should co-finance as some. That's very hard to do. Yeah. So right now, the only solution is to find third-party investment. Yeah, and we're not going via the venture capital road. It's usually through longer-term investments of family offices and so forth to drive this. Yeah. But I do see that the need for these alternative products becomes more important. Yeah, because the end users have no alternative. There is no palm oil available in the European market, right? And the motivation for here is really economic development. It's not necessarily that you need to change your existing industry. It is really protect the biodiversity and your resources that you have now. I'd rather have a field of rice than a field of oil. It feeds more people, right? With that in mind, still providing output, keep what you have, and make it more efficient. And I think this is where technologies like this become important. But it is international collaboration that can only implement this. We in Germany alone won't implement this. You in Indonesia by far will not implement this. But together with big companies, we can, with the right framework conditions, there needs to be tax incentives that need to be implemented to change. This is the case for Germany, the EU, and Southeast Asia. Yeah? And the money and the big companies are not necessarily here in Asia. They're in Singapore and in Hong Kong. Yeah.
Thank you. Any comments? Oh, okay. Another question, maybe? Okay, please.
Thank you so much for the insightful presentation. My name is Sarah Tulsa, and I'm currently working on my research project. Maybe this question is quite different with our topic today, but I know that do you have any experience about NLC, structured prepared products?
It's a particle. Can you speak up a little bit?
Do you have any experience working on manufacturing prepared maybe by this product that bring to compound?
Structural career.
Yes.
Yeah, structural career. The question for local problem.
Yes, I use palm stearin as a solid lipid and rice bran oil as the liquid lipid to bring a back compound into the cocoa products.
No, we haven't tried this. This is the obvious side.
Thank you. Regarding this cocoa butter, this is very interesting because cocoa butter is very expensive. Which also calculate using the oils from yeast. Is it cheaper maybe?
Yeah, it is certainly it is cheaper. You can imagine it's a matter of scale. At the moment, we calculate this at 10,000 tons, which is a mid-sized production facility. If you scale this and make it bigger, 10 times, your price will drop twofold. Yeah, so certainly it's something called economies of scale. A new technology is always expensive. Yeah, you need to find an economical framework to make it break even with the existing product at the beginning. And that's the key. You can produce tax incentive and lower regulatory barriers. In Germany, we are not very good with translating things from academia into industry because we are over-administered. As regulatory really is a barrier for innovation. And here in Asia, this is much lower. This is why the Chinese, for example, are so fast to develop new products, don't have regulatory barriers. Also less administration. And I think it's a chance for us also to transfer technologies here, because it's like a laboratory for new innovation. Yeah, you can just try it if something works, and then roll it out globally. Yeah, and it's just different regions in the world deal with innovation very differently. In Europe, unfortunately, we are over-administered. Most of the good companies that are startups from any country in Europe, they go to the US. Easier access to capital, lower regulatory and administrative barriers. Here in Asia, still, depending on the market, you still have a lot of regulatory barriers, for example, in cosmetics. Yeah, the cosmetics regulatory framework in China and Korea is far higher than Europe. Yeah. But that's where the money is. So, but in the food industry, certainly, I think we do have a large opportunity for collaboration. And to structure this really demands government collaboration. Yeah. And Indonesia joined the EU Horizon Research 3 years ago now. Yeah, so that this is an opportunity for collaboration because we have investment programs for flagships for projects where companies can come together and build new factories. And they get funding by the EU, and it doesn't matter where the factory actually exists. And for countries in the region that deploy certain to Indonesia information, because you're low-income countries, you are eligible for funds.
Thank you very much. I think the time's up. Is there any other question? Okay, no more? Okay. Okay, I would like to make a closing. Okay, thank you very much, Professor Brück, for the presentation. Yeah, this presentation gives us the perspective of the role of microbial biocatalysts and synthetic biology to address the transition toward the more sustainable and circular bioeconomy. Thank you very much. Please give one more applause for him.
Thank you to our moderator and also to our respected speaker for the example session. I would like also to express our sincere appreciation to Professor Dr. Thomas Brück for sharing such valuable knowledge with us today. As a compliment for our presentation, we would like to present a souvenir from the Master's Program in Food Science and Technology to Professor Brück. Thank you again to the moderator, our speaker, and all participants. Maybe before closing, we can take the commentation together. Dear participants, may you come forward to the face of together. One, three, two, one. Once again, 3, 2, 1. Is it a sign? Three, two, one. Once again, 3, 2, 1.
Oh, great.
Just feeling something there.
Yeah.
Yeah. Do you want?
Very important.